Okay, hello everyone. So I'm Ken Hoehn. I'm an assistant professor in the Department of Biomedical Data Science at Dartmouth. And today I'm going to talk about my work in inferring B-cell lineage trees from single cell and bulk B-cell lineage data. So B cells are a critical component of the adaptive immune system because they're the cells that create antibodies. Antibodies are proteins released into the blood and mucosa that provide protective immunity by binding to structures on the surfaces of pathogens called antigens. While this is very important for protective immunity, it's also possible for B cells to produce antibodies that bind to self-antigens and cause autoimmune diseases. They can also bind to food antigens and cause food allergies. And it seems that B cells play a dual role in cancer. So B cells can produce antibodies that bind to tumor antigens and act as a kind of tumor immune response. And they can also themselves become cancerous by developing into B cell lymphomas. So B cells are a source of great power within the immune system, both for good and for bad. The reason we can study B cells using evolutionary methods and using phylogenetic methods is because B cell diversity is shaped through mutation and selection. So antibodies are originally expressed on the surfaces of B cells in a membrane-bound form called the B cell receptor. And they're composed of a heavy chain and a light chain, and both of them are involved in antigen binding. When a B cell is activated by binding to its cognate antigen, it enters into a germinal center reaction. And in that reaction, the B cell enters into a lymph node where it rapidly proliferates and upregulates enzymes that damage its DNA at the antibody encoding genes. Some of those mutations that are induced by this DNA damage improve binding of the resulting antibodies. Some of them decrease binding. And the mutations that improve binding are selected, so those B cells survive more, and the ones that decrease binding are selected against, so those B cells die. So we have this cyclical process of mutation and Darwinian selection that overall improves the binding affinity of the antibodies that are produced. It also produces what we would call a B cell clone, which is a set of B cells that relate to each other by descent from a common naive B cell. So this B cell in the bone marrow and this B cell in the blood are part of the same clone because they relate back to this naive B cell. The way we build B cell lineage trees is at its heart relatively straightforward. So say we have this process of affinity maturation where we have B cells that are proliferating, dividing, mutating, and being selected. If we sample these B cells and sequence their BCRs, we'll find not a random sprinkling of mutations, but a hierarchy of mutations, some of which will be shared by all sequences, some by only a few sequences, and many mutations that will be unique to each B cell sequence. We can use that pattern of mutations to infer a phylogenetic tree, which is a model of this process of mutation and selection. So the tips of the tree represent the sequences that we observe. The internal nodes represent our unobserved intermediate sequences, and the branch links represent the mutations leading from the germline to the tips. This is a more complicated B cell lineage tree, but ultimately it is of the same form. So the tips are our observed BCRs, and the branch links represent the somatic hypermutations going back to the germline. And the work that my group is really interested in deals with different parts of this slide. So if we have B cells that are sampled in different tissues or different cell subtypes, and they're both present in the phylogenetic tree, we can ask questions like, how do we use the tree to understand B cell migration and differentiation? Or if we have B cells that are sampled at multiple time points from the same individual, how can we use these lineage trees to understand B cell evolution over time? But today I'm going to focus on something a little bit more fundamental, which is how do we build better B cell lineage trees? And that brings us to single cell sequencing data. So as I said before, B cell receptors are composed of a heavy chain and a light chain, and both are important for antigen binding. Both mutate as part of this evolutionary process. However, historically, B-cell lineage trees have only been built with heavy chains. And the reason for that is bulk BCR sequencing separates heavy chains from light chains, and people tend to just focus on the heavy chain. However, with single cell data, we can now get paired heavy and light chain sequences for each individual B cell. And this gives us a much broader set of nucleotide sites to build our trees. And it brings up the question of how do we best integrate this new data form in order to build better B-cell lineage trees? And I should say this work was headed up by Cole Jensen, who is a PhD student in the Kleinstein lab at Yale. The overall design of this study is to perform simulations that replicate paired chain B-cell evolution, build lineage trees, and then see which method best recapitulates the true tree. So we start with true trees that were simulated through a model of B-cell mutation and selection. We use those trees to simulate sequence alignments of just a heavy chain or a heavy chain plus a light chain. And then we can use those alignments to build trees and compare them to the true tree. We can also use these alignments to test which methods best perform when we have paired heavy and light chains. As part of this, it's really important to accurately recapitulate the evolutionary process of paired heavy and light chain affinity maturation in B cells. And one biological feature that we took care to replicate is the fact that light chains evolve much more slowly than heavy chains in B cells. So here we have a data set of B cell clones from several COVID-19 individuals. So each of these dots is a clone. And the x-axis is the heavy chain somatic hypermutation rate. The y-axis is the light chain somatic hypermutation rate. And we can see that in each clone, the heavy chains are much more mutated than the light chains, which indicates that the light chains evolve more slowly. If we look at a subject who recently received an influenza vaccination, we see a very similar pattern. And in our simulations, we recapitulate that lower mutation rate in the light chain. There are a lot of different ways of building lineage trees, and we tested 8, which are all implemented within our R package Dowser. And these 8 different methods are separated into 3 fundamental different frameworks. The first is maximum parsimony, where we just find the tree with the fewest mutations. The second is single partition maximum likelihood, where we use a model of nucleotide evolution and find the tree that maximizes the likelihood of the data. Importantly, this treats heavy chains and light chains as the same gene. And then we have multi-partition maximum likelihood, which is very similar to single partition maximum likelihood, but it allows light chain genes to have separate branch lengths. And this can allow the light chains to evolve more slowly or more quickly, depending on the data. One result that was very clear is that using paired heavy and light chains very consistently improves the accuracy of the phylogenetic trees that we build. To measure this, we use the Robinson fold distance, which is effectively the edit distance in the tree topology from the true tree topology. So a lower Robinson fold distance score indicates a more accurate tree topology. And this improvement in accuracy was very consistent in both maximum parsimony, maximum likelihood, and multi-partition maximum likelihood models. Importantly, if we look at the average performance of all of these methods, every method that used both the heavy chain and the light chain did better than every method that just used the heavy chain. So using the light chain gives you a much better improvement than picking the best heavy chain only method. However, in those results, every heavy chain was paired with a light chain. So that was fully paired data. But in real data, we often have missing light chains. And missing light chains can occur due to dropout in single cell data or from combining bulk BCR data and single cell BCR data. So if we mix the 2 different sequencing modalities, we can have situations where some cells have both heavy chains and light chains, and some cells have only heavy chains. And this is done for a lot of reasons, but mostly because bulk BCR sequencing has a much higher read depth. And that can give us the ability to link clones from different compartments. To test this, we repeated our simulations, but we progressively removed light chains from our single cell sequencing data. So in these plots, The x-axis is the percentage of light chains that are missing in the simulations, and the y-axis is the distance from the true tree. So this point at 0 is our fully paired data, and these points over here are heavy chain only data. And all the points in between are progressively more light chains missing. And we can see that as we discard light chains across all of the methods we test, we get ever increasing error in tree topology. But we still have an improvement in tree topology inference, even if about 40 to 50% of the cells are missing a light chain. So we can still have topological accuracy improvement, even if many cells are missing light chains. If we look at edge lengths, or the branch lengths of the tree, things are very different. So here we've plotted the proportional error in branch lengths. So branch lengths represent the number of mutations that happen in the tree. And again, the x-axis here is the percentage of light chains that are missing in the simulations, and the y-axis represents the error in branch lengths. So the trees down here are too short, the trees up here are too long, and the trees here are just right. And one thing that's very clear is that maximum parsimony consistently underestimates the total number of mutations in the tree, and this bias becomes worse as more light chains are missing. Interestingly enough, single partition maximum likelihood has exactly the opposite problem. It overestimates the length of the tree, and that problem becomes worse as more light chains go missing. But multi-partition maximum likelihood models give us accurate branch length estimates, even if 95% of cells are missing a light chain. And this is explainable based on how these methods deal with missing data. So maximum parsimony, no sequence, no mutations, so it underestimates the number of mutations in the tree. Single partition maximum likelihood assumes that data is missing randomly across both chains. So it effectively overestimates the branch length because it assumes that mutations are missing at the same rate in the light chain as they are in the heavy chain. Multi-partition maximum likelihood is able to allow the light chain to evolve more slowly, and therefore it accurately estimates the branch lengths even if light chains are missing. So we recommend using multi-partition models when building B-cell lineage trees with paired heavy and light chain data, even if, or especially if some light chains are missing. So everything I've talked about is implemented in our R package for B-cell phylogenetics, which we call Dowser. So you can build B-cell lineage trees using heavy chains and light chains, or just heavy chains, as well as all of the methods that I've talked about. It also contains methods for analyzing B-cell migration, differentiation, and evolution over time, which I didn't talk about today, but have been detailed in previous papers. So overall, Dowser is geared towards simple and user-friendly B-cell lineage tree analysis. And it's named for the process of using a Y-shaped stick to make questionable geographic inferences. You can take from that what you will. So Dowser is part of Incantation, which is a start-to-finish suite for BCR and TCR analysis. It's this ecosystem of R and Python packages. It's developed and maintained by the Kleinstein lab at Yale, where I was a postdoc until recently. It's modular and adopted to new BCR and TCR data types for nearly 10 years. We have a very active user hotline email, as well as fairly extensive documentation and tutorials and vignettes for getting started with BCR and TCR analysis. So if you're interested in doing that kind of analysis with your own data, I highly recommend checking out Incantation. So just to summarize, phylogenetic trees and B cells use mutations to link cells to a common ancestor. We can now use paired heavy and light chains to build B-cell lineage trees. Every method that we tested was improved from using paired heavy and light chains. However, most gave highly biased branch lengths if some light chains were missing. So for that reason, we recommend using multi-partition models, especially if you're mixing bulk and single cell data. And if you're interested in doing this kind of analysis with your own data, I highly recommend checking out Dowser and Incantation. So thank you for listening. Thank you to the organizers for organizing this session. If you're interested in Dowser or Incantation, these are the links to it and my email and lab website. Big thanks to our collaborators at Yale, especially in the Kleinstein lab and the Hoehn lab. If you're interested in this talk in text form, check out our paper in Journal of Immunology, which is basically going through this talk. And if you're interested in B-cell phylogenetics with single-cell data more generally, we recently published a review paper in Trends in Immunology that details the whole process of doing B-cell phylogenetics from single-cell data. And also thank you to the NIH for funding all of this. So.
Thanks for a great talk. So as you know, there are some cycles inside the germinal centers, right? I wonder, can you capture these cycles after learning these phylogeny trees?
So I think potentially you would need to, I think, sample the germinal center directly. So in most of the data that is worked with in the field, it's from peripheral blood sampling. But I think with fine needle aspirates or direct germinal center sampling, I think you probably could capture those cycles within trees, but you would need very high resolution sampling.
Thank you.
Maybe if I may ask one quick question. This is, of course, very exciting in terms of methods development, and I know that you're working on applications. And then there was this paper from Shane recently that's looking at, vaccine response, antibody evolution after repetitive vaccination in monkey models and so on and so forth. And then you really can see this mutational process happening when the, antibodies maturing. So what do you think are exciting applications of Dowser and tracing clone lineage?
Certainly. I think one of the most exciting for me is in what you said in temporal analysis of B-cell lineages, where we have longitudinal sampling over time, and we can use these phylogenetic trees to detect which lineages are evolving over time and how they're evolving over time. Really, I think in this slide where so we can use these to detect evolution over time in response to, for instance, vaccines. But also if we have B cells that are sampled in different tissues, we can use that to understand B cell migration. So there are a lot of applications, particularly in autoimmune diseases, which tend to be multi-compartment, where you have pathogenic B cells developing in one particular tissue and then migrating to other tissues. So one clear example is myasthenia gravis, where you have B cells maturing in the thymus and seemingly developing autoreactivity in the thymus and then escaping out to the blood. So, but yeah, I think there are as many applications as there are B cell mediated diseases. So.
Hi. I'm curious about this phenomenon that used to be called original antigenic sin, and then people call it immunological imprinting, I think, more recently. So does this, is it like the memory clones, B cell clones, when they're activated, they have a more limited capacity for mutation, so they sort of get stuck trying to make the same kind of antibody against the new related antigen, and they can't affinity mature towards the new antigen?
I think that is one explanation. We've seen that in humans after influenza vaccination, you can definitely get some memory B cell clones re-entering into germinal centers and accumulating new mutations. It wouldn't surprise me if that was reduced. So if the capacity of particular memory B cell clones to re-enter the germinal center and accumulate new mutations is reduced compared to naive B cells. But yeah, I think that is one explanation. Another explanation is perhaps there's just less antigen available because we already have antibodies available to bind to it. Other explanations are maybe memory B cells are just less, that the selective process of affinity maturation is just less effective on memory B cells, or maybe those germinal center reactions are less able to produce effector cells like plasma cells. But to my understanding, it's still unclear what exactly causes original antigenic sin.